Hey everyone! So imagine this for a second. What if you could just sit down at a computer, type out a string of letters, hit print, and have a machine build the actual code of life, DNA? Well, you know what? That's not science fiction anymore. Today, we are going to dive into how scientists can literally write, copy, and build with the fundamental blueprint of every living thing on the planet.
But that brings up a really fair question, right? Okay, you can make a tiny little strand of DNA. So what? What is the big deal? Well, answering that question is exactly what we're here to do. Because this one capability, it has completely changed everything. Medicine, research, our entire understanding of biology.
So, to really get how this is even possible, we need to start with the field that makes it all happen. This is where chemistry isn't just for looking at biology, it's for actively building with it. Right, so that field is called chemical biology, and the most important word in that definition is application. This isn't just about watching molecules do their thing. No, this is about getting your hands dirty. It's about designing and building chemical tools to go inside a living system and tweak it, poke it, and even reprogram it. It's a super hands-on science. Here's probably the best way to think about the difference between chemical biology and, say, regular old biochemistry. A biochemist might take apart a car engine to figure out exactly how it works. You know, they're observing. But a chemical biologist? They're designing new tools to tune that engine up to make it run better, or maybe even give it a brand new function. It's all about the difference between just watching and actually doing. Okay, so if chemical biologists are going to do stuff with biology, they need to start with the instruction manual. And in life, that manual is written in DNA. This is seriously how scientists can write DNA from scratch. It's this beautiful, elegant bit of chemistry. Basically, you start with a tiny bead, kind of like an anchor. Then in a super-controlled cycle, you add the building blocks of DNA, the A's, T's, C's, and G's, one by one by one. Now, the really clever part is using these protector chemicals that make sure each new letter attaches exactly where it's supposed to. Once you've spelled out your entire gene, you just snip the finished DNA strand off the anchor, and boom, you've got a custom-written piece of DNA.
Now, having one single copy of a gene, that's cool, but it's not super useful. To do anything meaningful, you need millions, even billions of copies. You need, like, a molecular photocopier.
And lucky for us, that photocopier exists. It's a technique called the polymerase chain reaction, better known as PCR. It is, without a doubt, one of the most important inventions in modern biology. Full stop. It uses a heat-proof enzyme and cycles through different temperatures to make copy after copy after copy, turning one strand of DNA into billions in just a few hours. It is an absolute game changer.
And you know the story behind PCR is just wild? The guy who invented it, Kary Mullis, was not your typical buttoned-up scientist. He was this brilliant, super eccentric chemist who supposedly got the idea while just driving down the highway. It's just a great reminder that these world-changing ideas can come from literally anywhere.
And believe me, it was a world-changing idea. In 1993, Kary Mullis won the Nobel Prize for it. And today, from CSI-style forensics to medical tests like the ones for COVID, PCR is absolutely everywhere.
All right, so we've written our DNA, we've made billions of copies of it. Now it's time for the really, really cool part, using that code to actually build something useful, like a protein.
So how do you do that? Well, scientists basically hijack one of nature's most efficient factories, bacteria. Usually it's good old E. coli. They pop their custom-written DNA blueprint into the bacteria, and the E. coli's own cellular machinery just takes over. It reads the blueprint and starts cranking out whatever protein the scientist designed. But of course, in science, there's always a plot twist, right? When scientists first tried to do this, to make human proteins inside E. coli, they hit a major roadblock. It turns out that while the genetic code is basically universal, different creatures kind of have their own dialects.
This gets at a problem called codon usage bias. The DNA code is read in three-letter words called codons. The problem is, our human genes really like to use certain codons, like AGA, for example, that are super rare in E. coli. So when the bacteria's machinery comes across one of these rare words, it kind of stumbles. It doesn't have the right tools handy, and the whole protein production line just grinds to a halt.
So what did chemical biologists do? They came up with this amazing solution. They engineered a special type of E. coli called the Rosetta strain. They basically gave the bacteria extra gear, extra machinery that lets it understand those rare human codons. It's like they handed the factory a universal translator so it can read any blueprint you throw at it. Just brilliant. Okay, so we've solved the translation problem. Our little E. coli factories are busy making our protein. But that leaves us with one final challenge. How in the world do you get it back out?
Right. I mean, think about it. Inside that single bacterial cell, your brand new, super important protein is just floating around in this thick, messy soup with thousands of other E. coli proteins. Finding your one specific protein in there is, well, it's the ultimate needle in a haystack problem. You need some way to just fish it out.
And that is exactly what scientists designed. They add a tiny little extra bit of code to their gene, which puts a unique tag, a molecular fishing hook, right onto their protein. These tags are designed to stick to one very specific thing. For instance, a really common one, the His tag, sticks to the metal nickel like glue. So you can just break open the cells, pour the entire protein soup over some nickel beads, and only your tagged protein will grab on. Everything else just washes away. It is an incredibly elegant way to purify something.
So let's just recap this whole powerful process. Step 1, you either write your gene from scratch or copy it with PCR. Step 2, you add the little bit of code for your fishing hook, the affinity tag. Step 3, you pop that finished DNA into your translator, E. coli. Step 4, you let the bacteria do all the hard work of making the protein. And finally, step 5, you break the cells open, use the tag to fish out your protein, and you're left with the pure, finished product.
And this brings us right back to the beginning. What's the big deal? Well, this is the big deal. This whole process is how we make countless life-saving medicines. The vaccine for hepatitis B, it's made this way. It's how we mass-produce enzymes for everything from laundry soap to cutting-edge research. It's the engine behind new diagnostic tools. It is the fundamental technique we use to study the very machines that make our bodies run.
So when you step back and look at it, we have gained this unbelievable power. The ability to read, to write, to copy, and to build with the operating system of life itself. The toolkit is here, it's getting more powerful every single day, and it leaves us with the most exciting question there is. What should we build next?